In this last video, we'll review the entire process. I'll review with you using an overview slide, which if you can talk to yourself, you're probably in pretty good shape. This is the slide you want to be able to talk through on your own. Practice with this. Explain it out loud to someone. Then you know you understand it if you can do it well without notes. I'll walk through it once. Make sure you know this. We have a mouse represented up top because immunologists use mice for their studies all the time because their immune system is very much like ours. We have a cell-mediated response and a humoral response. Let's talk about the cell-mediated first because for the most part, we're going to need our CD4 positive cells on both branches. If you have a T cell with their T cell receptor on their surface, they're going to need to come in contact with MHC in order to be activated. For a CD8 positive or cytotoxic T cells, the TCR on these cells are going to have to recognize their epitopes from the antigen on class 1 MHC molecules. The CD8 on the T cell surface helps stabilize the interaction between the TCR and the MHC presenting an epitope on the other cell. When this interaction happens, the T cells are going to be stimulated to want to kill the cell that they're interacting with. But they're not going to be able to do that unless they get cytokines from a T helper type 1. How do you activate a helper T cell? Helper cells are CD4 positive. Their TCR has to come in contact with epitopes presented with class 2 MHC, which remember are on antigen presenting cells that are phagocytes or B cells. The CD4 on helper T cells is the accessory molecule that helps stabilize the TCR interacting with the MHC2 presenting an epitope. So MHC2 molecules presenting exogenous epitopes will interact with the T cell receptor of the T helper, and the helper T cell will be stimulated to then release their cytokines. If they are T helper type 1, their cytokines will activate the CD8 positive T cells, and they will undergo clonal expansion and self-stimulation, and they will kill altered cells by using that perforin and granzyme system. This is how to kill using a cytotoxic T cell. For the humoral response using B cells, the B cell receptor is just anchored antibody on the cell membrane, advertising to the world which type of antibody that B cell can make. The type in this case refers to the epitope it recognizes. Because remember, each B cell can only make one type of antibody. Ultimately, one of the 5 classes and recognizing only one specific epitope. There are many choices, but once a B cell is committed to a particular type of antibody, that is the only type of antibody they're going to make. If they come in contact with their exogenous antigens floating around, that's going to stimulate the B cell to respond. It's probably going to internalize that antigen and represent epitopes of that antigen with MHC2 that it can stimulate a helper T cell, because it's going to need that helper T cell that's been stimulated to release Th2 cytokines. Once it does that, the B cell is going to then be activated to undergo clonal expansion. Some of those are going to be plasma cells and actively secreting that type of antibody that recognizes the epitope they're first presenting to the T cells. And some of them are going to be memory B cells. Remember, antibodies have many activities that can undertake, but their goal is to get rid of the antigen. This is how you need to be reviewing for your exam. Talk this slide through with as many details as you can from memory. Make sure that you understand how to activate T cells and how to activate B cells and the ultimate result of activating these cells, either killing hosts, altered host cells, or eliminating antigen by making antibodies. Also know the different activities of antibodies. Your immune system is really complicated, but I hope that I helped you understand it a bit. Reach out if you have questions. Good luck assimilating this new information.